Okay, this particular video is on cell surface markers and red blood cell typing. Please refer to my Word document, cell surface markers and red blood cell typing, when you review this. The human body has approximately 210 different cell types, subsumed into 4 tissue types you may recall: epithelial, connective, nerve, and muscle. The standard human has somewhere between 70 and 100 trillion cells, but roughly 210 different cell types. On each cell, there are identification flags, identification markers. These markers are called clusters of differentiation, nicknamed CDs. These identify the cell for your immune system to look at them and determine what cells to attack and what cells not to attack. When the CDs primarily are on the cell membrane are glycoproteins or glycolipids. They would be glycocarbohydrates protruding from the cell membrane, either attached in the cell membrane to a lipid, glycolipid, or a protein, glycoprotein. But it's really the carbohydrate moiety that is what your immune system looks at to determine. As you can see in this picture here would be, let's say, right here would be a protein, a glycoprotein. Right there would be a cell surface marker, and that would be the carbohydrates. Each of these circles are monosaccharides covalently bonded to make this, which we'll call an oligosaccharide. When we look at the various cells, 210 different cell types, we can separate 209 cell types from one cell type. The one cell type would be the red blood cells. The other cell types would be all the other 209. When we look at the cell surface markers, all of the others, the 209, use something called the MHC, which stands for Major Histocompatibility Complex. Remember, histo means tissue, histocompatible. What this alludes to is like for transplants, to see if someone is tissue compatible, one heart going into somebody else, or a liver. The major histocompatibility complex. Okay, the major histocompatibility complex, the word by itself, the term by itself, means the set of genes responsible for putting the flags on. So when you say major histocompatibility complex by itself, it means the set of genes in the DNA. When you say the MHC proteins, which you'll see later in this course, that means the actual protein flags, glycoproteins, put on the cell itself. So the MHC, major histocompatibility complex, determines the MHC proteins, which would go on the cell membrane. Now that will be discussed more in the immune. Okay. The red blood cells, however, use a different marking system. There are 30 to 32 different ones. The two I will discuss are the ABO and the RH, primarily because they are the most antigenic. An antigen is a substance that can stimulate an immune response. We have really two kinds of antigens, we'll say, for this point of discussion: isoantigens and heteroantigen. Iso means same. An isoantigen is your own antigen in you. A heteroantigen is something not in you. Well, why do we call it an antigen? Because if I take a cell from me, which is an isoantigen that I'm not going to attack, but put it in you, you would attack it. So humanistically, it is an antigen. It's just not an antigen in me, but it is an antigen, thus termed an isoantigen. But place it in someone else, not compatible, it would be a heteroantigen. Okay? When I say they are the most antigenic, there are degrees of antigenicity, as I alluded to, like offensiveness. Someone may say something to you that's offensive, but you say, oh, no big deal. But someone may say something that really hits at your core that's very antigenic, and you're ready perhaps even to fight. So just like there are degrees of offensiveness, there are degrees of antigenicity. If you place the wrong ABO blood type, or RH, in a person, there will be a violent reaction generally because they are highly antigenic. But some of the lesser of the 30 to 32 red blood cell groups may not cause any problem. Not that the body likes them, but it's not enough to take on a fight with it. Generally, when we type and transfuse blood, red blood cells, generally they're only looking at 5 to 6 of the most antigenic. So you have like the RH, the ABO, the KEL, the KID, the Stewart, the Duffy, the MEN. There are various systems. Of course, all we will primarily discuss is the ABO and RH. Okay. Let's talk about a little genetics to explain these surface markers. Okay. You have genes to put these on there. An alternate form of a gene for a certain characteristic is called an allele. So let's say you had a gene for brown eye color and a gene for blue eye color, that's theoretically say, which is not exactly true because there's a multiple gene process for eye color. But if one parent handed you the blue and another handed you the brown, then you would look at that, and each one of those would be for eye color, so they would be alleles. Alleles are alternate form of genes for the same trait. Okay? So when we look at the ABO blood system, it's what we call a multiple allele system, in that there are more than 2 alleles for the trait. There's an allele for A, there's an allele for B, and there's an allele for O. So it becomes a multiple allele situation. When we look at the relationship between alleles, we can see that in some cases you can have what we call dominant to recessive relationship or codominance. Now there are other relationships beside that, but that's not important for this particular subject. When there's a dominant to recessive relationship, as one parent hands you the dominant and the other hands you the recessive, then what will be expressed will be the dominant. That is what we call the phenotype and the genotype. The phenotype and the genotype. The phenotype in genetics is what the world sees. The genotype is what you actually have. So if one parent, in the ABO blood system, for example, A is dominant over O. So if one parent handed you the A and another parent handed you the O, you would type as an A. That would be your phenotype. Your genotype, though, would actually be an A and an O. So your genotype would be an A and an O, but what you would type as is an A, because in this relationship, this is a dominant to recessive relationship. A is dominant over O. B is dominant over O. So in either case, if one parent handed you O and the other handed you A or B, you would type as the A or the B. A is co-dominant with B. So if one parent handed you A and the other handed you B, you would type as AB because you would have some A cell markers and some B cell markers on the red blood cell. That's directly out. So the only way really to get the O would be one parent handed you O and the other handed you O. That's essentially how that would work. Okay. What are the cell markers for the ABO? They are actually glycolipids. The glyco, being the carbohydrate, is what is protruding from the cell membrane. The carbohydrate are really oligosaccharides. Oligosaccharides are small carbohydrates with 2, let's say, to 10 monosaccharides covalently bonded together. In this case, though, they're generally 4 bonded together. The first 3, which are attached to the lipid, remember this is a glycolipid, are called the H substance. The H substance is kind of like a flagpole, we'll say, to which you put the terminal carbohydrate. The terminal carbohydrate is what determines what your blood type is. So, for example, if you had galactose as the terminal carbohydrate, you type as B. If you had N-acetylgalactosamine, you type as A. So it's that terminal one. If you do not have a terminal, which would mean you have the H substance, then you would type as O. That's essentially what the markers are. How do you determine the red blood cell type? We use the antibody-antigen system. Antibodies are produced to fight antigens, let's say. One of the ways that antibodies work is to cause clumping of cells, because there are many different sites on them, and they can come in and clump things together so that maybe a macrophage can eat it. Okay. The antibody will only work if you see the antigen. So, if the antigen is there and you place the antibody on there, it will cause clumping. So in this particular test that we could do in the lab, you would put 2 droplets of blood from yourself on a slide. But you would have already turned the slide over using a grease pencil and put an A on one side of the back of the slide and a B on the other side of the back of the slide. You would turn it back over, put a droplet of blood on the A spot and a droplet of blood on the B spot on the other side of the slide. You would then come in and put antiserum. Antibodies are located in your plasma. So therefore, if a person had A blood type, they wouldn't want to have A antibodies because you attack yourself. Vice versa with B. So what we could take are produced A antibodies and put them on the A spot. And then some produced B and put them on the B spot. If you got clumping on the A spot, that meant that you had A flags, A antigens. Then that would mean A blood type, provided that you didn't also get clumping on the B side. If you got clumping just on the A and not on the B, you would have A blood type. If you got clumping on the B and not the A, you'd have B blood type. If you got clumping on the A site and the B site, you would have AB. And if you got clumping on neither, then you would have O blood type. So that's basically how that particular situation works. RH, once you understand the ABO, the RH is much simpler because it's not as many genes for that. There are many kinds of RH genes, but if one parent gives you an RH gene and the other one doesn't, you are RH positive. So if both parents give you RH, then you're definitely RH positive. If one gives you an RH gene, you're RH positive. The only way to be RH negative is if both parents do not give you an RH, then you would theoretically be RH negative. How do you check for RH? The same way you did for ABO, but instead of using antibodies for the ABO, you would use antibodies for the RH. If there's no clumping when you place it on there, then you're RH negative. If there is clumping, then you're RH positive. So essentially when a person says, putting it all together, that they are O negative, what does that mean? That from the ABO side, they're O. From the RH side, they're negative. So it's the same way. If a person says they're B positive, from the ABO side, they're B, and from the RH side, they are RH positive. That is essentially how it works. Thank you.